Good evening, students. Yesterday we discussed up to antigens and other properties. Today we are going to discuss about immunoglobulins, antibodies, and antigen antibody interactions and immune diseases. Clear? So we will discuss one by one. The first one is a antibody. So antibodies means you know very well glycoproteins. Antibodies are a glycoproteins. Kindly note it, which is important one. Antibodies nature is a glycoproteins, which is also called immunoglobulins, but all immunoglobulins are antibodies, but all antibodies are not a immunoglobulin. Kindly note it. All immunoglobulins are antibodies, but all antibodies are not a immunoglobulins. Clear? So antibodies means already I gave a definition for to you. When that foreign particles enter into our body, our immune system first recognize that one and produces some substances against that antigens, which is known as a antibody. So this is a structure of antibody. It having the Y-shaped. It has stalk and two limbs. This is called as a stalk. This is called as a limb. It has two stalks and two limbs. Each tip of limbs are called antigen binding site. This one. This is called as a antigen binding site. The tip of limbs called antigen binding site. Antigen binding site also called as a paratope. So epitope where it is present. The two things are most important one. Epitopes are present in the surface of antigens. So epitope means a antibody binding site. Epitopes are present in antigens. So epitopes means a binding for antibody, because that antigen provides some surface for the binding of antibody. So epitopes present in the surface of antigens for binding of antibody. So epitopes are a binding site for the antibody. So similarly, here the tip of limbs called antigen binding site, which is known as a paratope. So paratopes are present in the antibody. For binding of antigens. So paratopes are called antigen binding site. That is an important neat question. Epitope, paratope, and glycoprotein. The three is the most important neat question. The tip of stalk is called FC. The tip of stalk. This is called as a FC. The tip of stalk is called FC. It only binds to the phagocytes or complements. The stalk of FC only bind the phagocytes or complements. And it having the two chains. What are they? It is having the two chains. What are they? One is two chains means one is a light chain, another one is a heavy chain. This is called the light chains, the shorter one. This is called as a light chain. This is called as a heavy chains. So heavy chains are longer one. This one. The light chains are a shorter one. So I will combinedly teach that about antibody with immunoglobulins because immunoglobulins are antibodies. So I will combine with antibody with the immunoglobulins. So immunoglobulins are glycoproteins formed because I just now I told all antibodies are glycoproteins. So immunoglobulins are glycoproteins formed in response to antigens and react specifically with the antigens. So formed in response to an antigens. So for example, the particular antigen is enter into our body means it due to that entry of that antigen only that immune system will be responsive and produces some antibody against that antigen. That antibody will be react with the specific antigen. Which antigen is responsible for the stimulation of that antibody? That antibody will be reacts with the next one is immunoglobulins are immunological missiles produced by the body which kills the pathogens. Which kills the pathogens. Immunoglobulins are found in serum, body fluids, and tissues. So where the immunoglobulins are present means serum. Serum means you know very well. If you are collecting that blood means allow for coagulate it. I mean that blood clotting. So after that clotting, that clotted blood will be settled down. That upper layer that is called as a supernatant. It is a yellowish in color. That is called as a serum. That other body fluids. Body fluids means a liquid-like things. That is called as a body fluids. And the tissues you know very well. So the immunoglobulins are found in the serum, body fluids, and tissues. Immunoglobulins are produced by the vertebrates only. So immunoglobulins are produced in the vertebrates. Vertebrates means you know they are synthesized by the B lymphocytes and secreted by the plasma cells. Immunoglobulins are synthesized by the B lymphocytes and secreted by the plasma cells. That thing also I think you know because you should know. Because two types of cells are available. One is a T and a B. B cells only responsible for the antibody production. So B lymphocytes, how it produces the immunoglobulin means B lymphocytes or a B cells will produce the plasma cells. Plasma cells produce the antibody. That thing I already explained so many times. I told to you. Clear. So that is also important on the B lymphocytes secreted the plasma cells. The plasma cells will secrete the antibodies. Clear. The structure just now I told Rodney Porter in the year of 1962 proposed the basic structure of immunoglobulin. Rodney Porter. He only proposed the basic structure of immunoglobulins. That immunoglobulins are exist in the two forms. What are they means? One is a soluble immunoglobulins, another one is a membrane-bound immunoglobulins. Soluble and membrane-bound immunoglobulins. Soluble immunoglobulins are found in dissolved condition in the blood serum and body fluids. Soluble means dissolvable. It can able to dissolve. That is called as a soluble. So that soluble immunoglobulins are found in the serum and the body fluids. And the bound immunoglobulins. Bound immunoglobulins are found in the surface of B cells. Bound means you know very well it will be bind with any another the particular thing that is called as a bound. So bound means it will be binded with any another thing, any another thing like a B cells or T cells like that. So that bound immunoglobulins are found in the surface of B cells. They are referred as a surface immunoglobulin, shortly called as a SIG, small letter S, caps letter I, small letter G. Surface immunoglobulins, because that's that immunoglobulins, membrane-bound immunoglobulins are present in the surface of the B cells. That's why which is called as a surface immunoglobulin, which is also called as a membrane immunoglobulin, MIG, membrane immunoglobulins, because which is present in the membrane of B cells. So membrane immunoglobulins, all membrane-bound immunoglobulins are present in the surface of B cells, hence which is named as a bound immunoglobulins or surface immunoglobulins or membrane-bound or membrane immunoglobulins. Clear? So like this is a BCR, B cell receptor. B cell receptor. BCR means a B cell receptor. Receptor means already told. Receptor means gives us some space for binding. This one. Lymph of stalk. That is called as a binding site. BCR. This is a antigens. When that antigens are comes enter into our body, at that time it will be bound to the particular antibody, like a lock and key mechanism. How the lock specific key will be bound with the particular lock, like that antigens will be bind to the specific antibodies. Clear? That epitope of antigens will be binds to the parrot of our antibody. Clear? So here that membrane, in membrane immunoglobulins are found. That's why it is called as a membrane bound. Clear? The surface of immunoglobulins found in the surface of B cell is called B cell receptors. Approximately more than 1 lakh BCRs are present in its surface. So what is the chemical nature of immunoglobulins? Means immunoglobulins are glycoproteins made up of two polypeptide chains. Two polypeptide chains. What are they? Two pairs of polypeptide chains. That each polypeptide chain is a linear sequence of amino acids. Sequence of amino acids. Some amino acids are attached to the sugar chain. Hence, immunoglobulins are called as glycoproteins. Glyco means a carbohydrate. Carbohydrates are sugars. That's why which is known as glycoproteins. I again explain more that immunoglobulins are glycoproteins, which is made up of two pair of polypeptide chains. Two pair of polypeptide chains. Each polypeptide chain is made up of a linear sequence of amino acids. So amino acids means polypeptides. Peptides. Amino acids are linked by the many peptides. That is called as a polypeptides. That linked many amino acids groups are called proteins. Clear? So polypeptides, many peptidic group. Some amino acids attached with the sugars, hence which is known as the glycoproteins. Clear? Already I told glycoproteins. Amino acids or immunoglobulins are made up of glycoproteins. Proteins. Clear? The next one is it having the two kind of chains. One is a light chain. One is a light chain. This one, another one is a heavy chain. Heavy chain. Light chains are shortly called as the L chain. The light chains are shorter and identical. This is this one. This is a shorter and identical. Identical means it is a similar one. It's look like a similar identical copies. That each light chains are made up of approximately 214 amino acid, 214. That each light chains are made up of 214 amino acids. Based on the amino acid sequence, light chains are grouped into two types. What are they? Means a HEPA and lambda. HEPA and lambda. This is called as a lymph. This is called as a stalk. This one, that stick-like structure is called as a stalk. That lymph will be present in the edge. The star. The next one, when immunoglobulins contains the HEPA chains, which is called as a K-type or K-type. In case that lambda chains are present, is called as a L-type. That here next one is a heavy chains. This is called as a heavy chains. Immunoglobulins has a two heavy chains. This one, the heavy chains are a longer one. That this two is called as a heavy chains. That heavy chains are a longer one. That heavy chains also identical like light chains. That heavy chains are made up of a huge amino acids like a 450 to 700 amino acids approximately. The heavy chains has a flexible region is called a hinge region, which is called as a hinge region. This one. Hinge means that joining places. That is called as a hinge regions. That is a flexible one. Like our toothpaste, the tail and head portions will be connected by the neck-like structure. That is called as a hinge. Like that in immunoglobulin, so I-shaped. There are also that joining places here. It is a flexible one. So that region is called as a hinge region. That heavy chains also 5 types. What are the means? Gamma, alpha, mu, delta, and epsilon. Gamma, alpha, mu, delta, and epsilon. Next one is N-terminal end and C-terminal end. N-terminal end means this one. This is called as N-terminal end. So one end of each chain is called amino terminal end, which is called as N-terminal end, amino terminal end, because here amino groups will be present. That's why it is called as N-terminal end. Opposite end. This is called as a carboxyl end. This is called as a carboxyl end. Another end is called as a C-terminal end, which is also called as a carboxy terminal end, because in one terminal, in one end is amino group will be present it. This is called as the N-terminal end. Another one is called as a C-terminal end, carboxy terminal end. Carboxyl group is present it. Then the chains, the 4 chains are connected by the intra-chain disulfide. The 4 chains, 1, 2, 3, 4. 2 light chains and 2 heavy chains are connected by the intra-chain disulfide bond. Otherwise, the 4 chains are connected by the disulfide bond. Here 2 sulfide group will be present. Here also 2 sulfide. Here also 2 sulfide. That's why that 4 chains are interconnected by the intra-disulfide bond. Most probably that heavy chain here, that heavy chains are linked by 1 to 13 interchain disulfide bond. Heavy chains are linked by approximately 1 to 13 interchain disulfide bond. Each light chain and heavy chain by a single interchain disulfide. This one, light chain and heavy chains are here that disulfides bond occurs between the light chains. Sorry, heavy chains only. Here that disulfide bond occurs between the light chain and heavy chain. Here also that disulfide bond occurs between the light chain and heavy chains. Clear? The next one is the constant and variable regions. Immunoglobulins also having the 2 regions, namely constant region and variable regions. That constant region is called as a C regions, variable region is called as a V regions. Constant region means amino acid sequence remains constant. This one. Refer the diagram. The constant region, the amino acid sequence remains constant. Amino acids are a constant in most of the immunoglobulins, which is called as a constant region. It will be located in the base of the stalk. Like this. This is located in the base of the stalk. The constant region formed of both light chain and heavy chains, like that. This is the constant region having the heavy chains and light chains. In variable regions, amino acid sequence over variable regions. Just now I told enter about intermolecular here. This is called as a variable regions. That variable region amino acid sequence shows a variability. It shows a variability. So keep it in your mind, ma. Constant. Already I told, see the literal meaning. So constant regions means already I told which is made up of glycoproteins, mostly proteins. Sugar, when it is attached, is called as the glycoproteins. So proteins are made up of amino acids. The amino groups are a constant, which is called as the constant region. Here in amino groups, that amino acids shows some variability, hence which is called as the variable regions, which is located, variable regions are located near the N-terminal end. Clear? So those are the important points of immunoglobulin structures. Immunoglobulin structures. The next one is what are the types of immunoglobulins, sir? This level is okay, ma. So already so many times I told you, immunoglobulins only I can teach 4 to 5 years. But I think that is not important for you, because in a Neet point of view, you must know about what's the meaning of immunoglobulin, what are the nature of immunoglobulins, the structure of immunoglobulins, and the types of immunoglobulin at basic level only. That is enough. The next one is types. Next one is types. So types of immunoglobulins. What are the types of immunoglobulins are present means 5 types of immunoglobulins are present. Mostly IgG, IgA, IgM, IgD, and IgE. Those are the 5 types of immunoglobulins. What are the types of immunoglobulins, ma? Those are the 5 types. What are they? IgG, IgA, IgM, IgD, and IgE. So we will discuss one by one. We will discuss one by one. The first one is IgG. So before that one, you should know what are the basic properties of immunoglobulins. Means immunoglobulins are a glycoprotein. This is the Y-shaped, most probably Y-shaped, which is linked by the polypeptide chains. Those are the 2 light chains and 2 heavy chains. That half-life period is 2 to 25 days. Contain the disulfide bond. They agglutinate the antigens. That immunoglobulin IgG. That just now I told what are the 5 types of immunoglobulins means totally 5 types of immunoglobulins. What are they means IgG, IgM, IgA, IgD, and IgE. First one is IgG. The structure of IgG is immunoglobulin G. That serum immunoglobulin G is a serum antibody. Immunoglobulin is a serum antibody. It is also glycoproteins. That all immunoglobulins are made up of 2 light chains and 2 heavy chains. That Immunoglobulin IgG which is made up of maybe 2-sulfate, 2-trisulfate environments, that is IG1 and IG4, IG2 and 10 to 13 immunoglobulins are present, means that is called as IG3. That valency of antigen binding site is a 2. Valency of antigen binding site is a 2. The IgG is the greatest amount in the serum. It is produced particularly during the secondary immune response. Kindly note it that IgG is produced at large quantity during the secondary immune response. Molecular weight around 150,000. The sedimentation coefficient is a 7s. The IgG is a predominant class of immunoglobulins of blood. Mostly that IgG is present in the human serum. IgG predominantly involves in the secondary immune response. Kindly note it, my important question. Just now I told IgG participate predominantly in secondary immune response. That IgG secreted in the breast milk and provides the babies with humoral immunities. That is also important question. That IgG secreted in the breast milk and provides the babies with humoral immunity. The next one is IgA. So IgA means IgA is a antibody. IgA also glycoproteins, which is also Y-shaped, which is also having the 2 light chains and 2 heavy chains. That IgA, serum IgA is a monomer. It is also structurally, that IgA is structurally similar to the IgG. That sedimentation coefficient also 7s. 7s. That IgGA is a secretory compound. IgA also support for the antigen antibody reactions. IgA also found to produce the immunity against tapeworms. IgA present in the Clostridium protects the baby from the intestinal pathogens. Important question, ma. IgA present in the Clostridium. Clostridium, yesterday I told to you. What's the meaning of Clostridium? IgA present in the Clostridium protects the baby from the intestinal pathogens. So, breastfeeding reduces the gastrointestinal infections in the infant by reducing the entry of pathogens from the environment. The next one is IgM. So IgM is the largest immunoglobulin. Kindly note it, ma. That is important one. That among that 5 immunoglobulins, IgM is the largest immunoglobulins, which is also Y-shaped. It having the 2 chains, light chain and heavy chains. Each chain is interconnected by the intradisulfate bond. Which is a pentamer structure. It is a pentamer structure. IgD, IgE and IgG. It is a Y-shaped one. Clear? IgA is a dimer. IgG, IgD, Ig is a monomer. IgM only a pentamer structure is the largest one. IgM, largest immunoglobulin, is a pentamer one. Pentamer structure. The serum level of IgM is a very low, approximately 5 to 2 mg per ml. That here sedimentation coefficient is 19s. The biological properties, it is the first antibody to appear the primary immune response. That IgMs involved in the appeared in the primary immune response. Primary immune response. That the most natural antibodies, such as a natural classification of ABO blood grouping immunoglobulins, anti-A, anti-B. Many of the natural antibodies to the microorganisms and heterophile antibodies are the IgM classes. So you know very well that B blood group, O blood group, that A blood group having the A antigens, B blood group having the B antigens, but A blood group having the antibody B, B blood group having the antibody A. So that antibodies are IgM classes, IgM classes, clear? The next one is IgD, immunoglobulin D. Also glycoproteins. Blood serum. Cells. The immune antibody activity, but IgD, which is found associated with the surface of B lymphocytes along with IgM, found to act as an antigen receptor. So IgT act as an antigen receptor. It will be combined with the IgM. Clear? It is found in the B lymphocytes. Similarly, IgD binds with the basophils and mast cells and activates that one. So basophils and mast cells are activated by the IgD also. The last one is IgE. IgE. So IgE is immunoglobulins. It is also monomer, which is also the Y-shaped, having the two regions and C regions, etc., etc. That all immunoglobulins having the same structure, two light chains, heavy chains, disulfide bond, etc., etc. but IgE is found in only traces or very low level in the serum. So IgE is present in the very low quantity in the serum because synthesized by the very few plasma cells in the body. So very few plasma cells only synthesize the IgE. That's why which is present in very low level in the serum. The persons with allergic conditions, its level may be 50 to 100 times higher than of a normal person. So when the allergic reaction is present in the particular persons, found in the persons, at that time IgE level will be increasing. Here the sedimentation coefficient is 8s. That IgE usually known as a skin sensitizing antibody. IgE usually known as a skin sensitizing antibody. That's which is involving in the allergic reactions. Similarly, that IgE major function, other major function is provides the immunity against the Helminth worms. It provides the immunity against the Helminth worms. It is also provides the immunity against the protozoa parasites. Also provides the immunity against the protozoa parasites. That's all about the immunoglobulins. In immunoglobulins, important thing is IgA, where it is present, where it will be transferred, and forms of immunoglobulins. Those are the most important NEET questions. You should read all things. Those are the most important things. So generally, immunoglobulins are involved in the agglutination, precipitation reaction, and the next one is that's all about that immunoglobulins. So now I completed the what is the meaning of antigens and what is the meaning of antibody. The next one is how that antibody will react with the antigens. Reacts with the antigens, which is known as antibody antigen interactions or antigen antibody reactions. The interaction between the antigens and the antibody is called as antigen antibody interaction or antigen antibody reactions. It is abbreviated as AG and AB reactions. That antigen antibody reaction is a basic humoral immunity and antibody mediated immune response. That antigen antibody reactions, what are the basic characters means it is a very specific one. Antigen antibody reaction is a very specific one. Specific means that like a lock and key. Just now I told. That's why it is called as a very specific one. If it's the structure is antigen structure is various one, means it does not bind with the specific antibody. The structural it shows structural similarities are most important. That one, then only that specific antigens will be fit with the particular antibody. Clear. So that's why I'm saying that antigen antibody reaction is very specific. That antibodies are react with only the antigens responsible for the synthesis. Antibodies reacts with only the antigens responsible for the synthesis. That binding takes place between the epitopes of antigens and the paratopes of antibody. Just now I told what is the meaning of epitope and the paratope. Epitopes are present in the antigens, provides the site for binding to the antibody. Similarly, paratopes are present in the antibodies, which is a site for antigens. Clear? So bindings are free but reversible. Antigen antibody bindings takes place at the surface of the cells because the surface only the epitope paratope will be present. So it takes place in the surface of the cells. The denaturation of antigens or antibody during the reaction. Here there is no denaturation in antigen antibody reactions. If denaturation means there is no reaction. So no denaturation of antigen and antibody during the reactions. So the entire molecules of antigen antibody will be take part in the reactions. The both antigens and antibodies participate the formation of agglutination and precipitation. Agglutination and precipitation. Antigens and antibodies are combined at various properties. Antigens and antibodies are combined in varying properties. Antibodies shows as individual properties. Similarly, antigens also shows the varying their properties. Does not shows the similar property, shows a variability. That's why I'm saying antigens and antibodies combined in varying the property. The forces acting between the antigen and the antibodies are non-covalent forces. Kindly note it, that is a important need question. The forces acting between the antigen and antibodies are non-covalent forces. Non-covalent forces. So this is immunoglobulins. Immunoglobulins are having the 5 types. What are the means? IgG, IgM, IgA, IgD, and IgE. IgE. That the 2 light chains and heavy chains are present in the all 5 types of immunoglobulins. That all 5 types of immunoglobulins are interconnected by the disulfide bond only. That constant region and variable regions are present in the all 5 types of immunoglobulins. Only it shows us some variations in HEPA chains or light chains molecules. Clear. So another important things are IgG is a monomeric one. IgG, IgD, IgE. The 3 things are monomer in form. That is an important question mark. IgG, IgD, and IgE. GDE. The 3 immunoglobulins are monomeric forms. IgA may be in monomeric or a dimeric. Maybe monomeric or a dimeric. You can see the diagram in middle one. So IgA is a monomeric or a dimeric. IgM is a pentameric one. IgM is a pentameric one. IgM is a largest immunoglobulin. The remaining where that IgE is transferred, where what is the role of that IgA? That remaining the functions alone. Yesterday I told. Kindly recall it. Clear. So that's all about the immunoglobulins. So next one is antigen antibody reactions. Antigen antibody reactions. So yesterday we discussed about the antigens. Now I explained about the antibody. So next, how the antigens are interacted with the antibody. Clear. So then only that antigens will be neutralized. So that antigens should undergoes the reactions with the antibodies. Clear. So that the interaction between that antigen and antibody is called as the antigen antibody interaction. Antigen antibody interactions, which is also known as the antigen antibody reaction, which is abbreviated as AG-AB reaction. That antigen antibody reactions based on the humoral immunity or a antibody mediated immunity, because the cell mediated immunity does not involving the antibody. There the cells, macrophages are mast cells only involving in that one. But in the case of antigen antibody reactions, the humoral immunity or antibody mediated immunity. Here the B cells, plasma cells are majorly involving in the antigen antibody reactions. Clear? That antigen antibody reactions are a very specific one. That antigen antibody reacts with only the antigens responsible for the synthesis. For example, the particular antigen is entered into our body means that antibody will be secreted for the specific antigens only. That secreted antibody will be react with the same antigens only. The specific one means like a lock and key mechanism, that specific antigens will be bind only the specific antibody. That is called as a specific one. So antigen antibody reactions are a very specific one. That the binding sites also, bindings takes place within the epitopes of antigens and the paratopes of antibody. Just now I explained in immunoglobulin section. The binding bindings are firm but reversible. That antigen antibody binding takes place in the surface of the cells. So once the cell is present means that surface only that antigen antibody reactions will be takes place. Here there is no denaturation of a antigen or a antibody during the reactions. There is no denaturation. So denaturation does not occur. Reactions will be takes place after that one, that antibodies will be denaturized. Sorry, not a denaturized. It will be neutralized. There is no denaturation of antigens or antibody during the interaction of antigen antibody. Entire molecules will be takes part in the reactions. Entire molecules of antigens, entire molecules of antibody will be takes place during the reaction time. Both antigens and antibodies are participated in the formation of agglutination and precipitations or precipitates or agglutinates. We will discuss later what's the meaning of agglutination and precipitation. Antigens and antibodies are combined in varying proportions. Similarly, the forces acting between the antigen and antibodies are a non-covalent forces. That is an important question. That forces acting between antigen and antibodies are a non-covalent forces. So I will explain. So that the particular while the antigen and antibody reactions will be takes place means it should possess some important things. What are they means? The first character is antigen immune complex. Immune complex means when that antigens are bind with the antibody, at that time it forms antigen antibody complex. That antigen antibody complex is called as a immune complex, where the antibody is present alone or a bounded form, that is responsible for immune. So when the antigens are bind with the antibody, it forms a immune complex like antigen antibody complex. Clear? The next one is the specificity of antigen and antibody. Specificity means just now I told antigens antibody reactions can be compared to the lock and key system. A standard lock can be opened by its own key only, as one antibody can react with the own antigen only. That antibody does not bind with the another antigens. That antibodies can react with only its own antigens only. This is called as a specificity. Next one is a binding sites. Binding sites means just now I told that antigens having that epitope, that is called as antigenic determinant. Antibody having the paratope, that is called as antigen binding site. So that bindings occurs between the epitope and the paratope. That antibody IgM is a multivalent. Valency means the numbers of binding sites, numbers of paratopes. Clear? So IgM only having the multivalency. Approximately 5 to 10 binding sites are present in the antibody IgM. Similarly, some antigens also shows so many epitopes on the surface of antigens. Clear? The next one is binding forces. Binding forces means the binding forces of antigen and antibody reaction is due to the three factors. What are they means? Closeness. The closeness is very important one. For example, the first antigens are come closer to the antigens. So at the time only the strength of binding is a greater one. When the antigen antibodies are very closely fitted, the strength of binding is a greater one. Similarly, when the antigens or antibodies are apart, at that time the binding strength is very low. So that both should be very closer. Then only that binding efficiency is a greater one. The next one is intermolecular forces. Intermolecular forces means that forces which operate the between the two macromolecules. For example, that forces means just now I told that non-covalent forces operates the antigen antibody bindings. That non-covalent forces are it exists in 4 types. What are they? Means electrostatic force, hydrogen forces, hydrophobic forces, and van der Waals forces. Electrostatic forces, hydrophobic forces, hydrogen force bonding, and van der Waals bondings. Those are the examples of non-covalent forces. Kindly keep it in your mind. That question is an important one. That question may ask in a NEET exam. Clear? The next one is affinity and avidity. That is also important things. Affinity means strength of bond between the particular epitope of antibody and a single antigenic determinant, means epitope. So affinity is the strength of bond between the paratope of antibody and the epitope of antigens. That antibodies are bind strongly to the antigenic determinant, which is called as a high affinity antibodies. Next one is avidity. Avidity means the strength of the bonds after formation of antibody complex. That avidity means the strength of the bonds after formation of antigen antibody complexes. So mostly in affinity, univalent antigens. Univalent means antigens having the single epitope. So that affinity will be occurs in the univalent antigens. Avidity, AVIDATY. Avidity means multivalent antigens having the many types of antigenic determinant. When which is injected into the blood, each antigenic determinant stimulates the production of particular antibodies. So automatically that that antigens will be bind with the antibodies. So avidity means the capacity of antibodies to combine with the multivalent antigens. Capacity of antibody to combine with the multivalent antigens, which is called as avidity. That level you should know. Avidity means the capacity of antibody to combine with the multivalent antigens. Affinity means binding capacity of antibody with the univalent antigens. So univalent means affinity and avidity means a multivalent. Clear? The next one is how we can detect. That is called as a detection of antigen antibody reactions. That means types of, and that is also called as types of antigen antibody reactions. Types means so many types of antigen antibody reactions will be takes place. Types of antigen antibody reactions we cannot visualize by our naked eye, but that antigen antibody reactions will be takes place. Clear? So but that antigen antibody reactions we can visualize visible magnification of, such as precipitation, agglutination, cytolysis, opsonization. So like during that end of that reaction result we can visualize it. So before that reaction we cannot visualize it, but end of the reaction result we can visualize the antigen antibody reactions. So many types of antigen antibody reaction is available, like precipitation, agglutination, cytolysis, complement fixation, flocculation, opsonization, immunofluorescence, and etc. That precipitation and agglutinations are present in your NEET syllabus. Although Just you know the titles alone. What are the types of reactions is available? Name itself having that answer also. That cytolysis. Cyto means what's the meaning of cyto? Cyto means a cell. Cytolysis means what's the meaning of cytolysis? Lysis means a rupture. Cyto means a cell. So cytolysis in the dissolution of a cells. When the RBC is lysed, the cytolysis is called as a hemolysis. RBC consists of the hemoglobulins. So in cytolysis, RBC is only lysis. It means that is called as a hemolysis. When the bacterial cell is lysis, that is called as a bacteriolysis. Cell is lysis, that is called as a cytolysis. So which is caused by the complement fixation between the antigen, between the antibody and cell-bound antigens. Clear? This level is enough. The precipitation and agglutination. So precipitation means first we will discuss one by one. The two things are important one. Precipitation. So precipitation means a precipitin reaction. Precipitation is called as a precipitin reaction. So precipitation is antigen antibody reactions between the soluble antigen and its antibody. So precipitation means precipitation occurs in the soluble antigens and its antibody, resulting insoluble precipitate. Again, I repeat, kindly note it. It is a precipitation occlusion reaction is important one because the two reactions are which is used to detect the antigen antibody reactions. To detect the antigen antibody reactions. So based on the results of precipitation and occlusion only, we can conclude it whether that antibody is present or that particular antigen is present or not. Clear? So that precipitation means antigen antibody reactions between the soluble antigen and its antibody gives a insoluble precipitate. This is called as a precipitin. Here that the antibody causing the precipitation. Here the antibody causing the precipitation, which is called a precipitin. That reactions of antibody with the BSA, bovine serum albumin. Mechanism of this is a mechanism of precipitation. The precipitation due to the formation of antigen antibody complex. What's the meaning of antigen antibody complex? When the antigens are bind to the antibody and the both will be combined together, antigen and antibody, which is called as a complex. Complex means more than one. That is called as a complex. Antigen antibody complex means antigens will be bind with the antibody, which is called as a antigen antibody complexes. That the antigens are a multivalent and the antibody is a bivalent. Antibody is a bivalent. You can see this is antibody. So many antibodies are bivalent. It having the valence is only the two. The two joining places will be there. That is called as a bivalent one. This is a dotted shader like this one. This is called as a antigens. Multivalent. More than two, which is called as a multivalent. Multivalent means that epitopes are present in the antigens more than two epitopes, which is called as a multivalent. So here antigen is a multivalent, antibody is a bivalent. So each antibody is a bivalent molecules can bridge between the two multivalent antigen molecules. Here, that this is on binding site, this is on binding site of antibody. So one antibody can bind to the two multivalent antigens. Two multivalent antigens. When the antigens and the antibodies are optimal concentration, that precipitation is a complete and large lattice is formed. And large lattice is formed. Clear? Like this. So when the antigen and antibody is binded, we can get it a precipitin. This is called as a precipitation reaction. This is called as a precipitation reaction. Precipitin will be formed. Clear. Examples, some immunodeficients are examples of the precipitation: single immunodeficient, double immunodeficient, radial immuno, electrophores, rocket immunodeficients. In it, maybe ask the examples of precipitations. Clear. Immunodeficients are examples of the precipitation. Precipitations. Next one is the agglutination reaction. Agglutination. What is the meaning of agglutination? So agglutination is a antigen antibody reactions. The antibodies of serum causes the cellular antigens to adhere the one another to form a clumps. The antibody of serum. So serum antibody causes the cellular antigens, forms a clumps. The clumping of a particular antigen and its antibody. So the clumping of particular antigens and antibody. That antibodies that causes the agglutination is called agglutinins. So which antigens causes the agglutination reaction? That antibody is called as a agglutinin. Kindly note it, ma. So agglutinin means nothing. Which antibody involved in the formation of agglutination? That antibody is called as a agglutinin. Agglutinin. Similarly, which antigens aggregated are called agglutinogens. The particular antigens include bacteria, viruses, RBC, platelets. Those are the antigens. When the red blood cells are agglutinated, the reaction is called as a heme agglutinated. That actually, if agglutination occurs under the with the RBC, which is called as a heme agglutination, because the RBC consists of the hemoglobulins. When the bacterial cells are agglutinated, that agglutination is called as a bacterial agglutination. So based on that antigens, it will names as a bacterial agglutination or maybe a heme agglutination or a etc. etc. So what is the mechanism of agglutination means here also. Here the agglutination is reacted with the RBC, heme agglutination. Agglutination brought by the linking of two antigens and antibodies. Here also antigens also similar to the precipitation antigens. Antigens are a multivalent one, antibodies are a bivalent one. You can see the diagram. Agglutination is brought by the linking of antigens and antibodies. The most antibodies are a bivalent one. Antibody can link the two adjacent multivalent antigens. That IgM antibody is a multivalent. Just now I told in precipitation reaction, IgM has a multivalent one. It contains 5 or 10 combining sites. That is also already I told. Just now I told in precipitation side. Hence, it can able to link a more number of antigens. That IgM antibody have capacity to make a clumps. That IgM antibody only can make a clumping formation more effectively with a less number of molecules than that of a IgG antibody molecules. So IgM only can able to make a high clumping molecules than the other immunoglobulins like a IgG. Clear? The univalent antibodies cannot form a clumps or a lactase. Hence, the agglutination will not occur. Means antibodies with a single clumping site or a single combining site. So IgM only can able to make a clumping. IgG also involving, but IgM only efficiently makes a clumping. Clear. Here also that reactions are takes place that antigen and antibody and forms a clumping. In precipitation, precipitin will be formed. In clumping formed means that is if clumping is formed means that is called as a agglutination. Agglutination. For example, ABO blood grouping. ABO blood grouping. Agglutination refers to the examination of clump formation when the particular antigens and its antibodies are combined, which is called as a agglutination. So ABO blood grouping, here that ABO groups or RH groups are involved in the agglutination reaction. ABO groups are RH groups because based on that RH factor only, we can easily determine whether it is a positive groups or maybe a negative group. We can easy to say A group, B group or O group. Whether the O group is a O positive or O negative, A positive or AB positive, AB positive or AB negative. So that positive negative we can determine with help of a RH factor only. That is also important question. So RH factor only determine the blood group, whether it is a positive or a negative blood group. Clear? So the typing of blood, drop of blood sample we can mix it and we can fix in the slide. So at that time, here agglutination is source. So we can add some anti-sera. For example, anti-sera A, the sample belongs to the A. If the sample is clumped with the anti-serum B, here the first one is a A. The clumping is occurs. The blood sample is clumped with the anti-serum A, which is known as a blood group. Here, first one. So clumping occurs only the A. That is called as a blood group. If clumping occurs in the anti-serum B, which is called as a B blood group, the sample is clumped with the both A and B, which is called as a AB blood group. And the clumping does not occurs. I mean, there is no agglutination means that is called as a O blood group. O blood group. Sir, how that's just now I told this is we can say A, B, or O. But we should know the positive or negative RH factor means additionally we have to draw the one circle. Another thing we have to add it, that is considered as a O1. So in case clumping occurs A and O, which is called as A positive. In case clumping occurs A and B and O, that is called as a B positive. There is no clumping in O. Clumping occurs only the B means that is called as a B negative. Similarly, the clumping occurs the all things A, B, and O means that is called as a AB positive. Clear? So clumping occurs only the AB. There is no clumping in O means that is called as a AB negative. In clumping occurs only the O. Others there is no clumping means that is called as a O positive. The clumping does not occurs in any one circle. Means that is called as a O negative. So O only determine the positive or negative. That is called as a RH factor. So blood group is example for the agglutination reaction. Clear? So examples may be asked. So agglutination precipitin is a detection of antigen and antibody reactions. What are the applications of that agglutination means RH blood grouping? We can identify viral test that through that agglutination we can identify the typhoid. Through the viral test. Viral test is also example of agglutination reactions. Viral ABO blood grouping. So we can identify the ABO blood grouping, RH factor blood grouping, typhoid 1. Typhoid fever also we can identify through the viral test. So all those tests will be done based on the agglutination reactions. Clear? Based on the agglutination reactions. Next one is immunodeficiency diseases. What's the meaning of immunodeficiency diseases? Immunodeficiency. So due to the defects in the immune system is called immunodeficiency disease. That is also present in your Neet syllabus. So immunodeficiency disease. I think not in NCERT. Surely it is present in the Neet syllabus. So the disease is caused by the defects in the immune system is called immunodeficiency disease. So defect of immune system. Defect of immune system. So resulting in the depression or hypofunction of immune system. Means low level function of a immune system. That immunodeficiency diseases are classified into the two types of immunodeficiency diseases. What are the means? One is a primary immunodeficiency. Second one is a secondary immunodeficiency. Primary immunodeficiency. Second one is a secondary immunodeficiency. The primary immunodeficiency means detect the immune system caused by the, we will discuss one by one. First one is a primary immunodeficiency. The detect of the immune system caused by the defective genes is called primary immunodeficiency, which is caused by only the defective genes, called primary immunodeficiency. What are the types of primary immunodeficiency? Means stem cell deficiency, because the defect is occurs in the hematopoietic origin. Hematopoietic origin. Then B cell deficiency, defects in B cell. D cell deficiency, defects in T cells. Combined B cell and D cell deficiency, phagocytic deficiency, complement deficiency. Complement deficiency. There, the particular gene is a defective one. Clear? So that due to that stem cell B or T cell or phagocytic or a complement deficiency, it can able to cause us a one or order of many diseases like a reticular digenesis. Like a reticular digenesis is caused by the primary immunodeficiency. And George syndrome, lazy leukocytes, hereditary neutropeniasis. Those are the important diseases caused by the primary immunodeficiency. Primary immunodeficiency. For example, reticular digenesis means primary immunodeficiency, which is also caused by the defective gene. Here, the defective gene causes a stem cell deficiency. Stem cell deficiency means results of the disease, complete lack of lymphocytes in both circulation and bone marrow. Here, there is no lymphocytes, means immunity will be reduced. The B lymphocytes, D lymphocytes are a very, very important one. So complete lack of lymphocytes in both circulation and bone marrow. This is called as a reticular digenesis diseases. Clear? The George syndrome means the thymus fails to develop. That is called as a George syndromes. Thymus fails to develop. There is no thymus, which is a thymus is a primary lymphoid organs. Yesterday we discussed primary lymphoid organs. The primary lymphoid organs are responsible for making the lymphocytes. There is no lymphocytes means there is no cells. Clear? Because thymus are responsible for making a T cells. There is no T cells means there is no cell-mediated immunity. Clear? So that is called as a George syndrome, means thymus fails to develop it. Clear? Like that, that is called as a immune diseases. The secondary immune diseases means the secondary immunodeficiency means a depression of the immune response caused by the many factors other than the geneticals. Clear? Caused by the secondary immunodeficiency means not a genetical one. It is caused by the another factor than the genetical one, like HIV, some viruses. AIDS will be caused by the HIV. That is an example for secondary immunodeficiency. And drugs. Drugs means you know very well that alcohol or a drug tablet or etc. Drugs. So continuously we are taking the drugs means at least at the time also the disease will be caused.
and X-rays continuously that the body cells or tissues are exposed to the X-rays means that X-rays cause some mutation in the gene level, which like a cancer also caused by the X-rays. Clear? And aging also due to the lack of deficiency of nutrients, viral infections and autoimmune disorders. Those are the examples, some important examples of a secondary immunodeficiency. So primary immunodeficiency are caused by only the genetic reasons or a gene level things fails to produce the particular cells. That is called as a primary immune response. So second one is the secondary immunodeficiency, means apart from the genetical factors like alcohol consumption, exposed to the X-rays and some viral diseases, that is comes under the secondary immunodeficiency. Clear? You should know in secondary immune these things in your syllabus. In your syllabus that SCIT and AIDS is present. SCIT and leukocytes are defective. It cannot able to move, which is called as a lazy leukocytes. That is also important. Kindly note it, ma. Lazy leukocytes means leukocytes are defective. They are unable to move, which is called as a lazy leukocytes. Clear? So next disease is a severe combined immunodeficiency disorder. That is called as a SCID. Severe combined immunodeficiency disorders. It is also important one more. The SCID is a primary immunodeficiency disease caused by the defective B cells as well as in the T cells. Defective occurs in B and T cells. That severe conditional disease where the death result is 1 or 2 years. So severely, if it is severe one means within 1 or 2 days, death will occur. That humoral and cell-mediated immune response are depressed because it affects the B cell and T cell. So the both cell and humoral mediated immune response will be depressed. The patient who is suffering suffered from the SCID, that patient is highly susceptible to the infection. Who suffered from the SCID, the thymus is very small. The patients can be treated with the transplantation of bone marrow. So what is the remedy for SCID, sir? Means the transplantation only. So the transplantation means a bone marrow transplantation. Why, sir, we transfer the bone marrow instead of that other organs? Why we are not transferring that other organs like a leg or a hand? Because bone marrow only consists of the stem cells. That the center part of bone marrow consists of the soft tissues, which is called as a stem cells. The stem cells only have capacity to produce the whole organs. Clear? Like a totipotency. For example, if you are cut any plant, it means that plant can able to grow it due to the totipotential effect. Like that in humans, mammals also having the pluripotent. That pluripotent is present in the stem cells. That stem cells are present in the center part of the bone marrow. So that bone marrow transplantation is a treatment for the patient of SCID. Clear? The SCID is an important one. The next one is AIDS. That is also present in your syllabus. AIDS. AIDS means AIDS is a epidemic viral disease. AIDS is a epidemic viral disease of human population, which is caused by the HIV, which is caused by the virus HIV. This is a common one. It's easiest, but it is also important question. The HIV AIDS is a acute immunodeficiency syndrome, which is called as AIDS. That is also abbreviation. Also, you should know acute immunodeficiency syndrome, which is called as AIDS, which is caused by the human immunodeficiency virus. It's shortly called as a HIV. It is a acute immunodeficiency deficiency syndrome. The AIDS is caused by the infection of RNA viruses and lymphocytes, because the HIV is a RNA viruses. Because some people are thinking virus means RNA, a lot like that. That viruses, in some viruses, DNA also act as a genetic material. So mostly that RNA act as a genetic material. The DNA virus is also available, but HIV is a RNA viruses. In HIV, RNA act as a genetic material. In the AIDS, the T helper cells are depressed. T helper cells are depressed, which leads the suppression of immune system. Hence, the name is called as a immune deficiency. Here, that immune deficiency occurs. Who suffered from the AIDS? That person's immune system is suffered. That's why it is called as a immune deficiency. That immune system is depressed. That individuals susceptible to the infection and series of diseases developed until his death. For example, that person is suffering from that AIDS means he cannot tolerate the common cold also, because there is no immune system. That immune system is present means actively present means it will be fight against that common cold viruses. Clear? So here there is no immune system, because that immune deficiency is AIDS is an immune deficiency syndrome, not a disease. It is a immune deficiency syndrome, not a diseases. Here there is no immunity. So the common cold also cannot tolerate the person of AIDS. As a result, disease develops until his death. That initial infection of virus paves way for the development of complex diseases is called a syndrome. So what's the meaning of syndrome? Development of complex of disease. So AIDS is first discovered in America in the year of 1981. Now the disease is commonly found in all countries. HIV. This is the structure of a HIV. The AIDS is carried by the RNA viruses called human immunodeficiency viruses, which is abbreviated as a HIV. It is class of a retrovirus. That is an important question. It is a class of a retrovirus. It was discovered by the Luke Montagnier in the year of 1983. Very first found in America in the year of 1981. So that is comes under the retrovirus groups. It is a spherical in shape. HIV is a spherical in shape. Spherical in shape. It consists of an envelope, a capsid, and a RNA. HIV consists of an envelope called capsid and RNA. HIV has an envelope. That's why which is called enveloped viruses. That envelope is made up of a lipid bilayer. Maybe ask that HIV has a envelope which is known as enveloped viruses. That envelope is made up of a lipid bilayer. That envelope is studded with the spherical glycoproteins. That also spherical glycoprotein also studded in the lipid bilayers at specific regular intervals. That capsid is made up of a protein subunits. It's called as a capsomeres. What is the capsomeres in HIV means? That capsid made up of a protein subunits is called a capsomere. The HIV consists of a 2 RNA strand. You know very well that RNA is a single stranded one, but HIV RNA genetic material is a 2 RNA strand. And RNA is associated with the 3 types of enzymes. That RNA is associated with the 3 types of enzymes. That RNA is associated with the 3 types of enzymes, namely reverse transcriptase, protease, and integrase. RNA is associated with the 3 types of enzymes, namely reverse transcriptase, protease, and integrase. The 3 enzymes also important one. HIV also infect the T helper cells, macrophages, and monocytes. HIV infects the T helper cells, macrophages, and monocytes. The three things are important for the cell-mediated immunity. The three things are important for the cell-mediated immunity because monocytes and macrophages are cells involving in the phagocyticons. Clear? So it is affect the cell-mediated immunity. Those are the all things are important points. Kindly note it. When the virus enters in the nucleus of host cell, here uses it reverse transcriptase and converts its RNA into the DNA. You know very well what's the meaning of transcription. In molecular ingredients you have studied. In transcription means from the DNA to RNA. We can get the RNA strand from the DNA one, which is called as a transcription. So reverse transcription means that is a reverse action. That RNA we can get it RNA from the converts the RNA into DNA. This is called as a reverse transcription. That process is done by the reverse transcriptase enzyme. That's why when the HIV is entered into our body, that RNA molecules, genetic material becomes the DNA one. So at that time our immune system is confused. So because our human beings also having the DNA as a genetic material. So RNA, which is entered into our human body, RNA also becomes a DNA due to the action of a reverse transcriptase enzyme. Clear? So that's why our immune system cannot identify that material as a foreign one. When it is detected as a foreign one, at that time only it can able to destroy that one. So it cannot detect it because when that RNA is entered into our body, at that time that genetic material will be act as a own materials. So own materials cannot destroyed by our immune system. Clear? In this way, that RNA viruses of HIV will be escaping from our immune system. The same time, HIV also destroy the T cells, monocytes, macrophages. Those are the reasons cannot cure the AIDS. That viral DNA is integrated with the host one. After that, it will be integrated with the host DNA molecules. When the host DNA replicates, automatically that HIV DNA also replicates, and it will be produce the most number of HIV DNA molecules because that RNA, that host DNA and RNA DNA, HIV DNA, because that HIV virus's RNA becomes a DNA. That HIV viral DNA also integrated with the host DNA molecules. So both will be integrated. Normally, our own DNA will be undergoes a replication to produce multiple copies. At that time, that HIV DNA also replicates along with the own DNA molecules and produce multiple copies of HIV DNA. That viral DNA produces a mRNA. New viral particles are also synthesized. They come out lymphocytes and infect the new cells. So those is the important one. Your school book, NCERT books also, they elaborately given about HIV viruses. So that's all about it's how it is transmitted means how it will be transmitted. How it will be transmitted. That thing I think you know very well. Sexual contact. Both homosexual and heterosexual. The sexual contact. And the blood transfusion. Blood transfusion. Blood transfusion means one person is suffering from that HIV means suffering from AIDS means that blood is not checked. In case that not checking that blood, if you are transferring that one means in this way also that HIV virus will be transmitted from the one suffered person to the unhealthy person, another healthy person. That is called as a blood transfusion. And mother to fetus through the placenta. In case that mother carries the after zygote formation, mother carries the AIDS. At that HIV will be transferred to the mother to infant through the placenta. Tissue transplantation. Tissue transplantation means some organ transplantation or other some tissues transplantation side. So in case that donor is suffered from the HIV viral diseases, AIDS means at that time the transplantation occurs. The donor is a patient means at that time if transferred to the healthy person, there also HIV will be transmitted from the patient to the normal healthy person. That is called as a tissue transplantation. Injection with the unsterile syringe and needles. Injection. If one injection is the injection used to the patient of AIDS, the same injection is unsterilized. Before unsterilized, the same injection is used to the normal healthy person means in this way also that HIV will be transmitted. Injection with unsterile syringes and needles or etc. Those are the mode of transmission. In this, that is also important one. Sexual transmission, sexual contact, blood transfusion, mother to fetus through placenta, tissue transplantation, injection with the unsterile syringes and needles. Those are the mode of transmission one. Mode of transmission one. So what will happen after the HIV transmitted to the human being means the WPC will be reduced. T cells also affected. So how HIV will, AIDS will be treated means anti-retroviral therapy, prevention of viral replication, inactivation of reverse transcription, reverse transcriptase enzyme of RNA, stimulation of immune systems, crafting of bone marrow. Those are the some treatments. The treatment also completely cannot cure the AIDS. The treatment we can follow it. How we can control the AIDS means prevention means just now I told that syringes, needles and other materials should be sterilized and use it. HIV infected patients should be kept in scheduled places. Similarly, that HIV blood should be given during the blood transfusion. Treatment, just now I told antiviral and etc. etc. Antiviral therapy is called as ART. That is also important question. ART means antiviral therapy. Treating the HIV infection with drugs. The drugs does not kill the virus, but to slow down the growth of virus. Just now I told we cannot completely cure it. So while we are giving the drugs for HIV, at that time slow down the growth of virus. Hence the disease projection is slowed. Antiviral drugs are called ARV. ART means antiviral drugs. ARV means antiretroviral drugs. Clear? Those are the treatment of HIV. The next one is autoimmune diseases. The time is times up. So next tomorrow we will discuss about autoimmune diseases. Already I told why I am telling about tomorrow's topics means today you after completion of my class you should revise that one today's class, and tomorrow class autoimmune diseases means autoimmune diseases or diseases. After that what are the topics are present in the chapters. You should collect some information regards the tomorrow's topics also. Then only easy to understand it, because in school study is entirely different from the competitive exams and college study. So now that simultaneously you can practice how to study in college like. So because it will be useful while you are going to join a college. Clear? Okay, today class is over. Tomorrow we will discuss about autoimmune diseases and cancer and drugs, addiction, etc. etc. Thank you, students. Thank you very much.